Question number 1. What is deviation? Answer: a deviation is any unwanted event that differs from the approved processes, procedures, instructions, specifications, or established standards. Deviations can occur during the manufacturing, packing, sampling, and testing of drug products. Question number 2. What are the types of deviations in pharmaceutical industry? Answers: deviation is categorized as planned deviation and unplanned deviation. Planned deviation. Planned deviation mean pre-approved deviation. Planned deviation shall be approved before execution. Planned deviations are those the procedure are already planned and we know before they occur. Unplanned deviation. Unplanned can be defined as departure from written procedure. It may be uncontrolled event during performing the procedure or operation any stage of manufacturing, packaging, testing, holding and storage of drug product due to system failure or equipment breakdown or manual error. Question number 3. What is deviation categories? Answers: deviation can be categorized as minor, major and critical based on the impact on the quality. 1. Minor deviation. When the deviation does not affect or minimum impact on quality attribute, or critical process parameter, or an equipment or instrument critical for process or control, it would be categorized as minor. 2. Major deviation. Moderately impact on quality attribute, a critical process parameter, an equipment or instrument critical for process or control, the deviation is categorized as major deviation. 3. Critical deviation. A substantial potential quality attribute, a critical process parameter, an equipment or instrument critical for process or control, of which the impact to patients or personnel or environment is highly probable, including life-threatening situation, the deviation is categorized as critical. Question number 4. What are the types of drug stability studies? Answers: stability studies are mainly of following types. Long-term stability 25 degrees Celsius and 65% relative humidity. Intermediate stability 30 degrees Celsius and 65% relative humidity. Accelerated stability 40 degrees Celsius and 75% relative humidity. In-use stability. Question number 5. What is 21 CFR Part 11? Answers: 21 CFR Part 11 is the FDA's regulation for electronic documentation and electronic signatures. It outlines the administration of electronic records in a medical device company's quality management system and gives guidance for industry best practices. Part 11 was designed to cater to the evolving needs of the medical device industry, with the purpose of helping companies know how to use computer systems and software, particularly when it isn't working properly, maintain data safely and securely, and ensure data is not corrupted or lost. Ensure that approval and review signatures cannot be disputed. Trace changes to data. Prevent and/or detect falsified records. Question number 6. What are USP Chapters balances? Answers: Balance Chapter USP 41 and 1251. USP General Chapter 41, Balances, is mandatory and states the requirements for balances used for materials that must be accurately weighed. Weighing should be performed using a balance that is calibrated over the operating range and meets the requirements defined for repeatability and accuracy. USP General Chapter 1251, Weighing on an Analytical Balance, is a guideline applicable to balances used in all analytical procedures. It provides information on installation and operational qualification, IQ, OQ, performance qualification and balance checks, routine testing, minimum weight and balance operation. Question number 7. What is difference between calibration and validation? Answers: Calibration is a process or action that compares the measurement values of a measuring device or equipment against a reference standard and certifies the measurement accuracy. Validation, on the other hand, validation is a detailed documented process of confirming that the equipment or machine is installed correctly, operating effectively, and performing without any error. Question number 8. What is dissolution? Answer: Dissolution is the time required to release the active drug substances into liquid form in a dissolution medium under setup conditions, RPM, time, and temperature. Question number 9. What is USP chapter number for dissolution? Answers: USP chapter number 711. Question number 9. What is reference standard and working standard? Reference standard is also known as the primary standard. It may be derived from USP, NF, or other official sources, OR. Reference standards are authentic substances of high purity and are provided by the pharmacopoeial bodies. What is the working standard? Working standard is also known as secondary standard. An in-house standard is a substance of established quality and purity is shown by the comparison of a reference standard. Working standard is used for routine testing or analysis. Or a working standard is a standard that is prepared and qualified against a pharmacopoeial reference standard. Question number 10. What is the principle of HPLC? Answer. It is a technique used to separate the mixture of components based on the adsorption, partition, ion exchange, and size exclusion principle. Compounds are separated because the molecules move at different rates in the column due to different interactions between the stationary phase and sample. The molecules move at a different rate, therefore separation can be done.